Hello, Danny. Good morning.
Фу,
Wait,
зашел. Да, уже идет.
wait. We are already live on YouTube.
Hello?
Yeah, I can hear you. Can you hear me?
Yes, I can hear you as well.
Hello.
Hello.
Hello. Yes, Victor. Good evening.
Good evening.
How are you?
Доброго дня, шановні слухачі та друзі. Сьогодні буде проведений вебінар з професором Мухаммед Анжад Камалом з доповіді на тему Novel Global Community Educational Foundation and Interactive Conversation about Scientific Journey. Цей вебінар буде проведений у формі активної бесіди. Ви можете задавати будь-які питання у чаті професору, а ми будемо їх озвучувати. Тож приймайте активну участь. Приємного перегляду.
So, good morning, ladies and gentlemen. We are going to have a webinar about Novel Global Community Education Foundations and interactive conversation
Good
about...
morning, Professor Kamal.
Good morning.
So, if you have any question, feel free to write them in the chat because we are going to have an active discussion.
Are you listening my sound? My voice is clear?
Yes, the voice is clear.
Oh, okay, that's good. So we are waiting for audience. May I have a look how many audience are present at the moment?
Yes, at the moment there are 6 people watching, but there are people constantly joining us.
So no problem, I can wait if some people are late due to any reason, and we can maybe wait for a few minutes or whatever you suggest. I am not hurry. I can meanwhile doing my other things, replying my emails and some other things.
Yeah, I think that we should start because unfortunately there are a lot of speakers and our time is limited to an hour and a half. So if you don't mind starting now.
Okay, then other people they will miss, so it is better than half hour we can give them, because 1 hour is enough. So maybe we can start 6:30 of Sydney time and 9:30 will be your time. So then more people can join. I can do some activity, no problem. So I can engage these 6 people by different activities. Do you want to start with some activities?
Okay, so and what activities do you have in mind by that?
Okay, if I see the audience, then according to their choice, we can discuss. I can have interaction with the audience and then pick some activity.
So actually, I'm being told that there are more than 6 people watching us because we are.
Oh, 60. You said 60. 6 or 60?
Via Zoom, we have 6 people, and we have about 20 people watching us right now on YouTube live.
Oh, okay. Then okay, other people are watching YouTube. Okay, then I can start my Novel Global Community Educational Foundation, how it set up and how we started and from where it came, and some introduction about that one, and then coming back towards my own scientific journey from where it start. And if anybody, this is the first slide which last night I prepared quickly. So if anybody want to record my email, this is prof.ma.kamal@gmail.com, and my mobile number 61 is our country code of Australia, then 4693477758. So later on, if anybody want to have contact for collaboration, more information about our current special issues. At the moment, I am running more than 20 special issues in the medical science related journals, like a seminar in cancer biology, which has impact factor around 10, and some other like advanced Journal of Advanced Research, which has impact factor 7, and some other different nanotechnology and current medicinal chemistry, which has 4 point something. So different, the details of those medical science journals are already mentioned on one website, which I will show you later on. So, and about, so today in the talk map, there will be first topic about Novel Global Community Educational Foundation and background of some enzymology and inhibition of amidophosphoryl transferase, if there will be the time, because my scientific journey from where it starts from the enzyme and how I moved towards the imidophosphoryl transferase enzyme, and some brain facts, and then Alzheimer's disease, some facts and figures, and some enzymes like acetylcholinesterase and butyrylcholinesterase enzyme inhibition kinetics, then linkages of Alzheimer's disease with type 2 diabetes mellitus. So if there will be the time, I will move on those topics. Otherwise, I can stop at any stage, so no problem.
So I'm awfully sorry, Professor Kamal, but can you turn on your camera, please?
Oh, my camera is not turned on?
No, unfortunately it's not.
Okay. I don't know. So you mentioned this one from the beginning. Start video. Oh, yes.
Yes, we can see you, but can you put the camera? Yeah, that's great. Okay.
Is it better?
Yeah, that's better.
So, sorry for that one. I did not realize. It is good that you pick this point. So either I continue. What about my screen? Either my screen is there.
Yeah, we can see your screen, so you can continue.
Oh, okay, people are looking my screen.
Yes.
Okay, maybe take some feedback from the other audience who are watching on live from different places. So either everything is going fine on their side as well. They are also listening and watching the slides. Can you ask?
Everything is going fine, as we can see, as we can tell, because we have some people from our department watching as well. So if anything would go wrong, they would tell me. They would call me up and tell.
Oh,
so
okay. Okay, that's good. That's good. So these are some web links. So on this web link, if you go on this one, then you can see details of different special issues. So I'm trying to go on this one. Can you see this one? Special issues of different journals?
No, unfortunately, we can't see it because we can see your screen only on PowerPoint. We can't
see not another screen.
Yeah, so we can see only your PowerPoint presentation.
Okay. So is it possible if I send this link to you in the text and then you click that one on your side and open on your screen in the chat? Where is the chat option?
Yes, I think that's possible, or we can just send it. We can
then I can talk from here. So where is the chat options? It is not appearing. That one to send the text. Usually there. Yeah. Yes. Yeah. More. Yeah. Yes.
There are people writing in the chat, so you can send it.
Yes.
Right there,
Yes. So here. Yes, this one. So is it possible now? Did you receive?
Let me check.
Did you receive?
Yes, we can see it in the chat now.
Okay, now can you click that one?
So you want me to open that one, right?
Yeah, open. Yeah, please.
Okay, just a second. Yes, I did open it. So you want me to demonstrate it, right?
Yeah, like this one, not word by word, just little bit idea. So later on, if anybody want to come on this one, so first is like seminar in cancer biology. It is written in the blue, and then last date of submission, and then current medicinal chemistry. So if anybody is interested to submit their contribution, make sure that when go in the portal of the submission, then enter this hard code, which is highlighted with the yellow, BMC CMC 2020-HT-57. So this is very important. Otherwise, it will not come in our this special issue. So some like this one, Journal of Advanced Research, which has impact factor 7. So these are the details of different special issues which are at the moment open, and I am running this phytomedicines and current neuropharmacology 4.7, and So all the list is there. So if anybody is interested, or your professors are interested later on. And then on this website, there is my old some CVs type and some images, my achievements, which are related to scientific journey. So if anybody is interested later on, they can see these things. And these are the link for, can you see this link, PubMed?
No, unfortunately, we can't. Can you send it?
Okay,
Mail
yeah,
to
okay, okay, yeah. So I'm sending in this one. Did you receive?
Yes, we received it.
Yeah, now you can click this one, please.
Mhm. Yes, so just in a
So
second.
yeah, like in PubMed, if somebody write down Kamal-MA,
Mhm.
these are my list of publication. So only in this year, suppose if I write down 2021, so around we publish, it is appearing 30, but a few are maybe not belong to my team. So more than 20 so far in last 3 months we published it this year. So these are the list of this year publications. So this is of motivation and inspiration to the students. So today you are the student of medical students, and tomorrow you are the scientist of tomorrow. You are professors of tomorrow. So you will give lectures to our coming generations. So you can motivate, inspire them. You are the gliding star of the global nation. So therefore, it is very important to motivate and inspire our youth. Like one of our principal, when we were studying in the school, he was saying most of the time in morning time in the assembly that children of today, parents of tomorrow. So you will take over all these things. Anyhow, so I go back on slides. And then there is one other, this link regarding this one also. Maybe I can show you about Novel Global Community Education Foundation, which is the first topic.
Yeah, it would be also very great if you could send it to the chat as well, so other people.
Yeah. Yes. Yeah. Did you receive? You can click this one, please.
Yes, we received it as well.
Yeah, you can click this one.
Mhm. Yeah, so we received it. So what can you tell us about it exactly?
Yeah, in this one, I think I can. There is on the top right side, there is YouTube channel. Can you see YouTube channel on the top right?
Yes, there is a YouTube channel.
Yeah, you can click that one.
Yes.
Then maybe you can listen directly from our stars, our young members.
Okay, so I will send it to the chat as well. If anybody's interested in the YouTube channel.
Yeah, YouTube channel, they can listen. Maybe you can click one. You can click one and listen that one. How to stop this one? You can maybe click the first one too, and then if they want to listen, they can listen now.
Okay. I'll send it to them as well so they can listen. So I think that we should go on with our presentation. Can you turn it like full screen if that's possible? Because we see only the half of it.
This one?
Your PowerPoint presentation, because we see it
Oh,
only.
okay.
Can you open it full screen?
Yeah. So I want to get rid of this one. How is this? Oh, sorry.
That's way better.
So this is. Yeah, this one.
Yeah, so I think that we can share the web links at the end of our presentation, so everybody, all the other links, I mean, we can share at the end, so
Okay.
everybody can go and watch it there.
Okay.
Into it.
Okay, yeah, this one and then loop one. So these are my honors and awards. So it is related to now scientific achievements. Travel Fellowship and Sheikh Hamdan bin Rashid Al Maktoum Award in 2004. So started from 2006 and then in 2001 here in Australia. And then this was prestigious fellowship, U2000 postdoctoral fellowship awarded in 2000 by the University of Sydney, Australia, because there were around 283 candidates from all over the world. They selected 15 people and there was 10 referees panel. And it was on very challenging enzyme, amidophosphoribosyl transferase enzyme, which involved in purine de novo biosynthetic pathway. And that is very unstable due to iron sulfur center, and it oxidizes very quickly when we extract the enzyme from leukemia cell line. So that was prestigious in that sense, that highly competitive, and then they gave extra 25,000 grant for the setup of the lab. And these are my specialty: biochemistry, neuroscience, enzymology, toxicology, metabolomics, nanomedicine, manual lymph drainage. Do you know manual lymph drainage? You are medical students mostly. Lymph. One is our lymphatic system, lymphatic vessel system, lymphatic nodes, and manual lymph drainage for edema and in some cases stress and sinuses. So I also studied here this one in Australia and got the training how to move the lymph in the lymphatic vessel and lymphatic nodes. And if person has stress, so anti-stress also. And then leadership in managing staff performance, and also I studied chaplaincy. So chaplaincy is how to boost the spiritual level of the person when going into depression and anxiety without treating any medicines, only by spiritual way of whatever belief of the person. So then the responsibility, like nowadays, I'm leading global research team, more than 100 scientists around the world in the field of neuroscience research, neurodegenerative disorders such as Alzheimer's, Parkinson's, type 2 diabetes mellitus, and cancer. And we publish over 600 articles and 65 abstract international conferences. And this is my Scopus profile. This is a little bit old of 12 February. So maybe now it changed to 38 maybe. And this is a loop on the loop profile. And this is on the ResearchGate 56.51 ResearchGate score. And this is about enzymics. So enzymics, from where this word I derived from enzyme, the knowledge of handling enzymes and study enzymes and using enzymes for designing of anti-Alzheimer's and anti-cancer and anti-diabetic medicines, therapeutic agents. So generally enzymes are biological catalysts that speed up the rate of biochemical reaction without changing itself. So enzymes play important role in catalyzing many biochemical reactions for different purposes of life. So therefore, nowadays, most of the drug discovery research, they focus target on different enzymes to design such therapeutic agents, either inhibit or increase the rate of different enzymes. So therefore, my interest is mostly in enzyme inhibition kinetics. And then in 2000, when I got this U2000 postdoctoral fellowship, I started this amidophosphoribosyl transferase enzyme designing and mechanism of action of purine antagonist, while I had collaboration with Dr. Nigel Gregg in USA, who is the chief of neuroscience laboratory, in designing of specific inhibitor to inhibit either acetylcholinesterase like phenserine and tolserine, and semserines to inhibit the butyrylcholinesterase, which is the sister enzyme of acetylcholinesterase. So these are, they are trying to have in clinical trials different compounds for anti-Alzheimer's purpose. So enzymic, anti-stress, lymphedemic unit, anti-leukemic units. So these are the 4 things which I want to establish. And then this is a further introduction. I don't know either people are interested in enzymes. How many people are studying or working? Anyone is, can you ask from the audience, anyone is working or doing research or have interest in the enzyme side?
So yeah, we can ask them. So if somebody's interested, can you write it down in the chat? While Professor Kamal, you can continue, and if there are going to be any responses, I'm going to tell you. Okay?
Yeah, yes, yeah, because otherwise I will go very fast if people are not interested in, because these are more than 300 slides on enzymes. So like a structure of enzymes, it has 2 sites, active site and peripheral sites of enzymes, and they are specific shape like lock and key models, and how enzyme act and hydrolyze. There are 6 different classes of enzymes, and in active site, like suppose how catalysis is taking place and its binding site. So catalytic site and binding site to bind with the substrate, and then catalytic site it perform the catalytic action of the enzyme. And then there are the cofactors. There's 2 types of cofactors, organic cofactors and inorganic cofactors. So without the binding of the cofactor, the protein, enzyme protein is inactive, but when cofactor bind with the inactive protein, it becomes the active protein, active enzyme. So those L1, L2 substrate can then bind. And cofactors are 2 types, inorganic cofactors and organic cofactors. Like these are the type of organic cofactors, like in blood, heme, so prosthetic group, and like coenzymes, NAD, FAD, these are the coenzymes example. And then how it is apoenzymes, but when cofactor combine with the apoenzymes at the active site, then it become holoenzymes or functional enzyme. Substrate is the reactant in biochemical reactions which bind with the enzyme, and then enzyme change substrate into the product. So like this is the enzyme. So when join with the substrate, join with the active site of the enzyme, so then catalysis is taking place. So intracellular enzyme and then extracellular, which are outside of the nucleus. And then the importance of the enzyme in medical research, in the cancer research, neurodegenerative disorders, diabetes, and cardiovascular diseases. So these are so many importance of the enzymes, how enzymes are used in designing of different medicines, and anti-inflammatory agents, microbial therapeutic enzymes, and their roles in different digestive and enzyobiotics and anti-cancers and different field. So microbial enzymes, nowadays people are working a lot on microbial enzymes as well, deriving from different microbes. So this is about my inhibition of amidophosphorybosyl transferase by new antifolates, design and mechanism of action of purine antagonists. So this was very challenging work in University of Sydney back in 2000. So this is the background of that enzyme and importance of enzyme inhibition kinetics. Like I50 is the basic parameters. 50% inhibition is taking place, that concentration of inhibitor which cause 50% activity of the enzyme. And then there is other like Michaelis-Menten constant Km and Ki inhibition constants, so different kinetics. So this is the Australian Chemical Institute. I am also chartered chemist, fellow in the beginning member, and then now I'm the fellow member as well of Royal Australian Chemical Institute, and also Australian Institute of Medical Scientists. So further importance of the enzyme in anti-cancer drugs. And then my goal to discover new effective medicine for most rapidly rising diseases that include metabolic syndrome, diabetes, cancer, Alzheimer's, Parkinson's. So these are the different, this is our so people will not get bored. So this is something related to Sydney. So do you know what is where is this place in Sydney? Have you been someone here in Sydney?
Unfortunately, no, we haven't been there.
Okay, when you will come, then visit this one. This is Opera House, very famous tourist place, Opera House. So there is very high amount of ticket for different entertaining programs here, and different functions in different halls here, Opera House near the. And this is our University of Sydney, where I done my 3 years postdoc of working on inhibition of imidophosphorybosyl transferase enzyme for the development of anti-cancer drugs in the supervision of Professor Richard Christopherson. So this is a very famous point of the Vice Chancellor office in our University of Sydney. So by this way, you will feel that you are sitting here in Sydney. This is my Professor Richard Christopherson. Now he recently retired and became an emerite professor. So this is the general information about cancer. Hopefully everyone know about the cancer and then its treatment, different strategies and complementary therapies. And this is ginseng in Chinese medicine they are using, and loss of how the normal cell growth and then cancers in the case of cancer cell divisions, and different naming of cancers, different kinds of cancer, population-based studies, in which country more cancer. Antifolate are the oldest of the antimetabolites class of anti-cancer agents. And this is the background of MTX and AMT, which I used during my postdoc. So chemical structure of folate, which deficiency can cause anemia. And these 3 important antifolates. Inhibition of imidophosphorybosyl transferase in cure of leukemia. So estimation of the precursor of nucleic acid in the presence of antifolates, anti-leukemic. This one, yeah, it can come down. Yeah. Therefore, in our study, the focus was on primary inhibition of the early de novo purine pathway. So we use the radiolabel protocol for optimum hot intermediate of the de novo purine biosynthesis produced in the human leukemia cell line. CCRF stem cell was developed by me. And this is nucleic acid, 2-type DNA, RNA, nucleotides, purine, and pyrimidine bases. So these are 2-way. One is de novo way, and one is the salvage way. And nucleotide and deoxynucleotides are required by all living cells, and their metabolic pathway, inhibition of the pathway by biosynthesis of the nucleotide phosphate block transcription, and consequently the proliferation of the cells. So this was our aim. On this basis, the requirement of rapidly dividing cells for synthesis of NTP as a precursor of nucleic acid has led to the development of selective antifolates as candidate for chemotherapy of leukemia. So how assembly of IMP is taking place through 10 bridging reaction. So in the presence of glutamate, so DHF, how convert into other compounds, so dihydrofolate, dihydrofolate synthase, and all these in the presence of different glutamates. So these are the 10 reactions of purine biosynthesis pathway. So the first one starting from the phosphoribosyl pyrophosphate to phosphoribosylamine in the presence of glutamine. So in this one, this is catalyzed by imidophosphorybosyl transferase enzyme. And this was my focus, how to inhibit this one. So it was very challenging task because it denatured very quickly. So in the presence of glycine, aspartate, glutamine, carbon dioxide, and phenyl tetrahydrofolates and ATP, this compound IMP is formed. And this IMP, which is further going into the either DAMP, deoxy adenine monophosphate, or deoxyguanone monophosphate. This was the award I received by Sheikh Hamdan bin Rashid Al Maktoum Award for original research paper published in the Emirate Medical Science Journal. So they gave, I think, $7,000 and ticket from Sydney to Dubai and business class and their VIP status. This is the first committed step which I mentioned in the previous slide, that how glutamine and phosphoribosyl pyrophosphate in the presence of substrate, how convert into phosphoribosylamine, and in the presence of amidophosphoribosyl transferase enzyme and glutamate and inorganic pyrophosphate. And this is then further it can be divided into two parts. One is reaction is taking place on N terminal and other is on C terminal of the enzyme. So on the N terminal, glutamine is catalyzed, and on the C terminal, phosphoribosyl pyrophosphate in the presence of NH3 converted to phosphoribosylamine and inorganic pyrophosphate. And pharmacological importance of these enzyme inhibitors in cancer, in arthritis, in inflammations, and microbial infection. And this is about the assay, how it was a challenging task how to assay this enzyme. First, I tried spectrophotometric methods, inorganic pyrophosphate method, then one other inorganic methods, then radioactive compound I used, glutamate, and then radioactive glycine I used. And this was more successful as compared to first these three. And challenging is because this iron sulfur center, in this enzyme, there is this iron sulfur center which oxidizes in the presence of oxygen and denatured whole enzyme active site within few seconds. That was very challenging task. So this was one presentation I was giving here in the University of Sydney. So this is how optimize the enzyme. Those who are interested in the kinetics, they can understand much better. So all this is now kinetics, standard curve for inorganic phosphate, standard curve for bovine serum albumin, and partial purifications, and then activity versus the protein. So this is also Michaelis-Menten constant, how to determine Michaelis-Menten constant, and growth curve of CCRF stem cells versus time, days. So doubling time, how to calculate the doubling time, and then how to standardize the glycine on HPLC by using the radioactive detectors and UV detector. So all this is, this is TLC by just 0.5 macroliter, and at different times 0.5, 6.5, 10, 20. By increasing, then this was product separation was taking place, and then determine this one, its quantity, and then plot it. So this is again kinetic study. So this was the assay which I developed, phosphoribosyl pyrophosphate from the cell extractor, and then orotidine, and then enzymes, and then doubling enzymes, orotidine decarboxylase. And then 1 microliter I plotted on the plates, TLC, and separated the products on this one, and then determines the quantity. So this was one other touch for health. Kinesiology. Who knows about kinesiology? Anyone know about kinesiology? In your medicine or related to medicine, somebody studied kinesiology? No one in the audience? So here is the Australasian College of Kinesiology. So I attended their workshops. So this is, yes, this is a standard.
Maybe when we have time in the end, you can go a little bit further about kinesiology and tell us something about it.
Why not? I can do the activity about related to kinesiology. Just 1 minute. It very brief activity. If people are okay, so
Yeah.
you can follow me. You can follow me. So everybody become active. So just follow me whatever I do. So you will also do the same thing. So like put your left hand on your tummy button, and your right hand like this thumb and index finger on your upper lip. On your upper lip, and then rub the upper lip like this 3 times. Now change the position. Right hand on the tummy button and left hand again 2 fingers on same upper lip and 3 times. And when you do this one, have in the mind that mindfulness screening along with this, that how our creator created and gave us the structure of lips and the teeth which are under these lips and the function of the lip which you studied in your human anatomy and physiology, and the function of the teeth which you studied in your anatomy and physiology, and the density of the bones and all this sharpness and the whiteness and how teeth are joined with each other and how nerves are connecting with the teeth. So have imagination of all those things. So then after that, this collarbone, and then you make your 2 finger and 1 thumb like this, right hand, and left hand again on the tummy button, and this one, then rub your collarbone just behind the collarbone 3 times, and now you think all the human physiology and anatomy which is related to this part and the importance of this part, which is the connection through the terminus. Here are the terminus points. So connection with through the terminus point, connection of brain, head, inside the head, the brain with the spinal cord and all the parasympathetic system. So connection of sympathetic system with the parasympathetic system. So in that way, you can think, and 3 times you rub like this one, and then change the position, and then again with the left hand 3 times. And then third step is you can, your right hand, you can on your back, your tailbone. So in this position from top to the bottom, and then you think all the sacrum, your sacrum, coccyx, your sacrum to coccyx, and lumbar region, lumbar spine, lumbar region. So you think that how the small bones in their structure and how these fused and what is their role in our sitting, in our standing, in our walking, and then change the position. Right hand, again tummy button, and left hand on the sacrum and coccyx, and then move up and down like this position, oval position, and then think all the anatomy and physiology related to this part of the body and its importance. And then the last thing is in this activity, you can now think, you can stand up if you can. Everyone stand up. Yeah. And then you think that I am getting all the positive energy. I am getting all the positive energy which have been created for the service of humanity by the creator. And then I'm taking it, absorbing it in my mind, in my heart, in my lungs, in my digestive system, wherever your hands are going, in that organ. And now I'm excreting all the negative energy, jealousy, hatred, every negative things out of my body. And then I am now free of all negativity, and I am positive, and I am ready to serve the global humanity based on sincerity. Done this activity, everyone? How do you feel now?
Great. Actually, we feel ourselves much better.
Sorry, I didn't get your answer.
It was great, actually. I think that a lot of people enjoyed this, including me and Victory. We enjoyed it very much.
Yeah, it
Oh,
was
good.
okay.
Okay. Any other from audience? Anybody want to give feedback?
So
Yeah, someone gave his feedback. I think, oh, Anastasia said is better. She feels positive.
Yeah. Now you can have last one, long breathe in, like long breathe in, hold, and then release. And then one other, okay, maybe later on. So I think we have no problem of time. So how much time left?
we have around 45 minutes left.
Oh, okay. So okay, I'm going quickly. Maybe we did not receive any response when we asked from audience that who is interested in enzyme.
We actually had. Chris is told that he is interested in, and he is happy to hear about it. So if you can go any further about it, audience will be happy to hear it.
Oh, okay. So this is how to standardize radioactive. Nowadays, I don't know how many people are using radioactive material in research on your side. So anyhow, this is good information. So, and these are the different compounds which we collected from USA and Europe. And I tested on amidophosphorybosyl transferase enzyme and how much was the timings, accumulation time of the substrate phosphoryboside pyrophosphate by inhibiting the enzyme, and the time, and how much was the fold, how much fold. So on that basis, then I designed this compound as a novel compound to test, and like theoretical compounds which can inhibit the amidophosphorybosyl transferase enzyme. And I shared with one lady, okay, I don't want to give her detail. Anyhow, because I did not receive any fundings after 3 years, so it was not very useful for me. So she was in USA, I think. I gave to her that, okay, you continue the work, but then I don't know what she done or not, and what is the progress of this one. And I was thinking to make it patent, but I could not have any fundings to make patent and go further. So then these are the results of different concentration and different those molecules which I showed in the previous slide. And they are cellular phosphoryboside pyrophosphate accumulation at dual concentration of the antifluoride 0.1 and 1 micromolar concentration. And this is also cellular level in the cell in the presence of control, which mean the absence of these compounds, and then in the presence of these compounds, methotrexate, pretaxinil, lamotrexol, PY899, PT523, MTA, metporin, PY873. So different, they have different accumulation. So this is a fold of accumulation, and then here is the time versus accumulation. And these are also related to that one. Then I did some kinetic study of this one. Then interaction of methotrexate and lamotrexol, an enzyme of de novo purine biosynthesis. And this was like a HPLC on how HPLC works, to dual piston pump, gradient controller, column, one UV detector, other radioactive monitors, and how I extracted the leukemia from leukemia cell line metabolites in the presence of those 10 compounds, and then centrifuge, washing, and centrifugation, supernatant, neutralize, and then centrifuge to get the filtrate, and then load on this column of HPLC, and then elution of metabolite by using 2 buffers. And then these peaks came of different metabolites. And this is also in the presence of radioactive compounds, human leukemia cell grown in different condition of incubation time in the absence and presence of methotrexate. So by increasing the incubation time, like this is only 5 hours for the control. So GTP, ATP, ADP, and this is in the presence of methotrexate. So in the methotrexate, look, is methotrexate because inhibit, therefore there is no formation of GTP, ATP, ADP, so they block. So then the increase the period of 16 hours, so more ATP here, 5 hours, this much ATP, and then 16 hours, so more ATP produced, and then 64 hours, more incubation of glycine, and that glycine is showing us the and then these are also HPLC results of the control UV, and then this is radioactive in the presence of carbon-14 glycine. These are UV, and these are carbon-14 glycine, and this is for methotrexate, Lomotrexol, and MTA. This is our house. She is my youngest daughter.
That's a sweet photo. Very cute.
And so these are also different results in different way. So then we use the azathioprine as well. So in the presence of azathioprine, methotrexate, so control azathioprine and azathioprine plus methotrexate to see which pathway has been stopped. And then to further determine small peaks of different metabolites. So this is control PTX 1 micromolar antifolate. So here concentration, we change the concentration and effect is also different. So like this is control, and then in PTX, all these peaks reduce, and in Lomotrexol, methotrexate, MTA has no effect. So it is similar like a control. So, but these three, they inhibit different pathways. So this is the effect of PT523 on GAR synthetase, which is the second, catalyze the second step. And this is IC50, we determine that this is activity versus concentration. And by this way, we determine the IC50 value by two different ways, by percentage activity versus percentage inhibition. And then 1 over V versus concentration to determine the Ki value, 53 micromolar. This is also SAF orotidine, phosphoribosyltransferase transferase, standard orotidine, OMP, UMP. And this is in silico study. We also did in silico study, computational study, and published that one. This abstract and schematic representation of de novo, and then sequencing my collaborators from Pakistan and from different part of the world. They done this study. And this is one other study, Sidra Batul, they are also from Pakistan. So they done this study, glycinamide ribonucleotide transformylase inhibition by PY873, PY899, and derivative of isophytic acid. So this is also computational in silico study. So overall, we developed a thin layer chromatographic assay for phosphoribosyl pyrophosphate extracted from human leukemia cells. And steady state level of phosphoribosyl pyrophosphate was 102 micromolar measured in CCF stem cells. Leukemia cells growing in culture have been exposed to a number of antifolates, and their effect upon cellular level of phosphoribosyl pyrophosphate was determined. A mechanism of action of methotrexate and Lomotrexol on de novo purine biosynthesis in human CCRF stem leukemia cells was elucidated by using HPLC. A level of nucleotides synthesized in the de novo purine biosynthesis indicated that both antifolate induced depletion of ATP, ADP, AMP, GTP, GTP, IMP, and NAD in the treated human leukemia cells. So this opened a new avenue for the assay of amidophosphorybosyl transferase, catalyzing the first step of the de novo purine biosynthesis, conversion of phosphoribosyl pyrophosphate into phosphoribosylamine. And by this way, we established a road for designing of new antifolates. So all mono, di, and triphosphates of N-formyl glycinamide ribonucleotide were also decreased by methotrexate and lamotrexide in the presence of azathioprine. When I showed in the previous slide azathioprine, when we used azathioprine, so then these mono, di, triphosphates of N-formyl reduced. It means inhibition is taking place due to presence of azathioprine before that step. So these results support that two tested antifolate induced primary inhibition of the early de novo purine pathway. Inhibition of amidophosphorybosyl transferase catalyzes the first committed step of the de novo purine pathway, and focused on the inhibition of this rate-limiting enzyme catalyzes the first committed step. This research is of critical and immediate relevance to the development of novel selective antifolate type inhibitor as an effective new class of anti-cancer drugs. And the role of natural compounds in cancer treatment. There are a lot of papers on this topic. The medicinal plant, how they have different anti-cancer, anti-diabetic, antioxidant, and characteristic of plant-based. So molecular carcinogenesis and targets involved during tumorigenesis, and the role of nuclear factor kappa B in normal and cancerous cells. And this is our UTS. After University of Sydney 3 years, then I was working here in Sydney. There is UTS. UTS means University of Technology Sydney. University of Technology Sydney. It is maybe 10-15 minutes walking distance. This is the huge building, walking distance from Central Station of Sydney. So this was a lab at UTS where I was working. So this is the tower building of UTS. So these are the projects which we submitted in King Abdulaziz City for Science and Technology in Saudi Arabia. I'm like a editorial board member of 41 journals. These are my collaborators, my best collaborator and the first collaborator, Dr. Nigel Gregg, who is the Chief of Drug Design and Development Section in the Laboratory of Neuroscience, Biomedical Research Center, National Institute of Aging, National Institute of Health in USA. And she, Professor Chin Kyo, here at UTS. And Mars Lariel, she's in Italy. And this professor and Dr. Chain Bano, now she moved to New Zealand, and she's still in Professor who are in Malaysia. So this is a long time ago, third regional training workshop on plant biotechnology, which I done before my PhD in Pakistan. And this is a letter of my U2000 postdoctoral fellowship, in which it is mentioned there were 283 candidates from 280 applicant for the 15 fellowship to be offered in 2000. So, and this is Australian Academy of Science. They also gave me Young Researcher Award in 2001 when I was younger than now. And this is scientific recognition in France and USA. And this is also in Greece. And this is my master. These are international awards. And acknowledgement to Professor Richard Christopherson, his lab, Dr. Nigel Gregg, Dr. Shinchin. This is once I presented here like a, I have different talent, not only scientist or professor, but I'm also entertainer. I can entertain, I can sing songs, I can play different other activities, anti-stress activities and other things. So like now we did the kinesiology. So this is here in Australia. Once they invited me in one Christmas party type function where they gave me the opportunity to express my views. So I said, pure mission, pure and pure mission, how to unite the global humanity. So I made a song on this topic and I sang and they liked that one. This is Dr. Wodo manual lymph drainage, which I was talking about. So I did the basic course and therapy 1 course. Dr. Wodo manual lymph drainage, how to move the lymph in the lymphatic vessel and lymphatic nodes for different pathological purpose. And this is our very famous Three Sister Mountain here in Sydney. And this is also the tourist place. When people are coming, they visit this one. There's some story also about this Three Sister Mountains. And this is about my PhD thesis. Effect of some anti-cancer drugs on human erythrocyte acetylcholinesterase enzyme. So how people are feeling?
Everybody is enjoying. You can continue.
So everybody's okay?
Yeah, everybody is enjoying. Everybody likes.
Are they? If they are sleeping, we can have one more activity. I can listen now from them. So why not one come forward, one volunteer, and or maybe 2 volunteer, and we can have like a drama type activity. One become patient and one become doctor. So maybe Victor and Alexander, one of you, or maybe one other on the back, become patient, like maybe dementia or Alzheimer's type or Parkinson's type or something, and then other become doctor, that how the person will approach, or maybe 3 people, one become the receptionist. So how person take the appointment to the receptionist over the phone or on the face, and then how the receptionist forward to the doctor, and how doctor, then I will give feedback that what you did and what it was supposed to do. So if you want 1-2 minutes activity, how it is?
So I think that we can try it. We have a person from the chat, so we've invited.
First ask from audience if anyone.
Yeah, we invited an audience. We have Christos here. Christos, can you hear us? Can you turn on your microphone? I don't know.
All right, so I guess the audience, like someone should know how to act a drama.
Yeah.
Victor, can you hear me?
Because you are a student and young, so not be bored. So when you participate, you involve, then it will be fun. So it will be not like a only dry science.
Just now we will see who else we can take. So if somebody wants to participate in our activity, can you write?
Yeah, I can guide further how to perform. But if first a person become ready and come forward, that who is ready, so then I can give the instructions to that person who will perform what role. So need 3 people, one receptionist, one patient role, and one is doctor role. So 3 roles. So who want to take these 3 roles?
I can take any role.
Okay.
Who are you? Who are you?
Christos. It's Christos, Professor Kamal.
Christos. Oh, Christos. Oh, okay. Yeah, Christos, you are here. Okay, that's good. So you are taking which role?
Anything you want. I can take the doctor.
Oh, okay, you become the doctor, but people can see you. You should be on the screen, so everyone
I
can see
think
you.
that I need the right to open my camera. I only have the right to open my microphone.
Oh, okay. That person who has the camera, so then everyone can see how they are acting.
So,
Is there any other person who has
so
camera?
is there anybody here who has the camera on?
No, I can open my camera, but I'm not designated in Zoom to open my camera. Like, if you can change this, if you can make me co-host or something like that, I will be able to do so.
Oh,
Okay, depend upon the Victory and Alexander. Is it possible?
Just now, I will have a look whether or not it's possible. Just a second.
So it is other test.
Hello?
Yeah, now I can see Christos. Welcome.
Yes, welcome, Christos.
Okay, so you are the doctor. Christos is the doctor. Who is the receptionist? Who want to be receptionist? Who receptionist mean in front of the doctor office on the front bench, who can take the information of the patients and then inform to the doctor or arrange the appointments and like that type of role.
so I think Alexandra can take the receptionist and I'll take the patient.
Okay. Okay. I will be there.
Okay, Alexander, are you happy?
Yeah, I'm happy.
Okay, so you are the receptionist and Victor is the patient. Victory.
Yeah.
Oh, sorry, Victory is the patient. So the protocol can be Victory can call, if it is over the phone, call to Christos and discuss about briefly the matter and what is the reason to take the appointment with the doctor, and then the Alexander will arrange the appointment with the doctor, contact to the doctor when doctor is available, and then finalize the appointment. And then when you will come and visit, then take you to the doctor, and doctor will then diagnose or discuss or recommend something or prescribe or test or whatever. And then you think whatever disease or your problem, or you can think and you can present that one, and how all of you are performing your duty. And audience are also watching you, all audience, is it right?
Yes, everybody's watching us.
Okay, then audience can also, maybe from audience there will be some judge, 3 people judge, so who can mark and give the feedback. So who can be the judge from the audience? Appoint 3 judge.
Okay, we can find the judges. Let's say, how many do we need? We need 3 or how much?
Yeah, three, like a 3 referee or 3 judge from the audience. Minimum 2, maximum 3.
Okay, so do they need to talk with us or they can write down their?
No, they can write down. They can write down, and if there is a possible to talk, they can also then express their feedback at the end.
Okay, so I can see that there is one girl who is willing to play. Her name is Anastasia. She can be the judge, but she can only write.
One should be also from your local language background person who is not understanding English, but what is Russian language or whatever is your local language.
Ukrainian.
So one person from there. So everybody has contribution.
Okay, and we have one more girl. Her name is Yana. She speaks our native language.
Okay.
She will also be the judge.
Okay, so one is Yona is the judge. How to spell Yona?
I can write it down in the chat for you.
Oh, okay.
That's how to spell her name.
Yeah, Y-A-N-A.
Yana, yes.
Okay, so who will be the second judge?
Oh, so actually the second judge, she
Will
told
not
us
be able to- yeah.
she's in the bus right now and she's watching the webinar from the bus and she has connection interrupts.
Okay, so only one judge it means?
So I
Yeah,
think
so we can try with one.
Okay, okay, no problem. That's fine. So go ahead. Is there any question? Is there any confusion
so,
for
Professor,
the
like,
role? Yes.
the patient is just going to pick any symptoms he wants,
Yeah,
right?
yeah,
And
yes,
add to the
yes.
doctor.
Yeah, anything, even the COVID or whatever. Yeah,
Dr.
anything.
Christos. All
Whatever.
right, please go.
So
Good morning,
flexibility.
Mr. Mdu. Nice to hear from you. How can I help you today?
Yeah, good morning, doctor. I don't know. I have
Stop.
the problem.
Please stop.
Okay.
So it should be not directly with the doctor. So patient will contact to the receptionist.
Receptionist. Okay.
Alexander is the receptionist. So you contact to receptionist and discuss about your matter and then arrange the appointment. And then Alexander is the receptionist who contact to the doctor to take the appointment. And then when you will come, then at your day of appointment, Alexander will introduce you to the doctor. So you don't have direct contact with the doctor. Is it right?
Okay.
Yeah, I guess it is.
So I'm a little confused there. Okay.
Yeah, go ahead, please.
Right. Receptionist, I'm calling you.
Hello, my name is Alexander. Nice to
Yeah,
meet you.
good morning. My name is Victory. Am I speaking to Ukraine Polyclinic?
Yes.
All right. My name is Victory. I just have this complaint. I don't know. I would like you to set me up with a doctor. I'm having a hard time remembering what I ate last week. Usually, that is like it does not happen to me, but these days I have a hard time to like remembering what I ate, and like there's a little confusion with the way I perceive things now. Like I'm just seeing different colors. Like I'm talking to you right now, but when I'm looking at my background here, I'm seeing different colors. It's not being like it's not specific color, and also I have this ringing in my ear, and I'm just confused. Like when can I see the doctor? I think I'm going crazy.
Okay, so let me help you up. I need to check when the doctor is available to see you.
Okay.
So, wow, you're actually in luck because he's ready to see you in about 3 minutes. He has free time. So I need to contact him and warn you about you coming. So can you please hold on the line for a moment while I talk to?
Sure.
Okay. I'm calling Dr. Christos. Dr. Christos, good morning. Can you hear me?
Alexander, how are you? Good morning. Yes.
So as I can see, according to our program, you don't have any appointments in
Indeed,
the next 5 minutes. Am I right?
Yes, indeed.
So there is a patient coming to you. Do you mind giving him a look?
Yes, if you have talked with him, can you give me a little of background, like what is the chief complaint, what kind of problem does he have?
Okay, so the most important thing that he has is a memory loss over the past 2 weeks, and he has problems with identifying the colors. So this is, as far as I understood, 2 main issues.
All right. Please tell me the name of the patient and of course send him in. I will be.
Okay, so the patient's name is Victory. So I can send him in, right?
Yeah, sure.
Victory, can you join us?
Oh, good morning, doctor. Thank you for seeing me.
Good morning, Mr. Victory. Welcome to the Ukraine Polyclinic. How can we help you today?
All right, sir. So like I told your assistant earlier, I said I have hard time remembering. And right now I have pain in my back. Like it looks like I'm turning around. Like I can't really hear very well. So like, doctor, what is going on? Like,
All right.
wait
a minute. I can identify 3 problems here happening at the same time, according to what you say. You have a memory loss, you have a problem in hearing, and you also have back pain. Can you please tell me which of them started and when? Like they started in the morning, they started 1 week ago?
Okay, so as I remember, I think it started this week. Like I'm having a hard time remembering like little details, like what I ate last week.
Hmm?
But doctor, I don't think it's memory loss because come on, I can still remember my name. Like, do you think I'm going crazy?
Don't worry. No, you don't. You're not going crazy. Of course, the overlapping health problems make you feel intimidated, but we will find a way out for sure. Don't worry. Just try to answer my questions as accurately as possible. Okay?
Okay, but doctor, what about the ringing I'm having? Can you give me something for this so I just go home? I'm tired. I don't want to stay in the phone for long. Like, can you prescribe me some meds so I just go home?
Of course. Let me first conduct an examination, and after that, I will be happy to prescribe you all the necessary medicine so you can go home and then back to your work or your studies or whatever. So right now you have these three problems. I guess that hearing loss and back pain started in the morning, and memory loss started 1 week ago. Is this right?
Yes, and
All
my
right.
age is 50.
All right. My assistant has already noted this in your file, so I have this information, but thank you for letting me know.
Okay.
So tell me, do you have a history of other health conditions?
Oh no, everything is fine. In my family, we are not sick. Everything is
Are you
okay.
taking any medication for any reason?
Medication, no.
Have you ever been hospitalized for any reason?
Of course, maybe one or two accidents, and also I think I have previous back surgery. So maybe it's the pain I'm feeling.
Okay, that's interesting. When did you have back surgery?
That was in my early 30s.
Early 30s. Okay, and you
Yeah.
again had a problem with pain in the back and difficulty in movement and walking.
Oh, it was just like a car accident. Like
Ah,
a truck hit me, so they took me in for surgery, and they had to do some surgery. You know, doctors do their thing at the
yeah,
back.
yeah, of course, understood.
Okay, yeah.
All right. Tell me something else. Anyone in your family had any problem with memory loss, like your grandfathers or your parents, probably?
Doctor, no, it's not memory loss. Like we're strong in my family. We don't do memory loss. Like everything is
Perfect,
okay.
perfect. I'm very glad to hear this.
Okay.
All right. Concerning the dizziness, how do you feel right now?
Like something is turning, like ringing.
Okay. Please look at me right now and try to follow my hand. Okay, with your eyes. No, not your head. Your head should be stable, but try to follow my finger only with your eyes. Okay. All right. Oculomotor functions are fine. Um, Normally, and if we were in reality, I would have to do a test called Dix-Hallpike. This is for dizziness, and this involves some specific kind of movements to define the cause of this. But for the sake of time and the workshop, we will say that we performed this test, and you have the benign kind of dizziness, benign dizziness of position. So this is something that we can easily fix with a medication called Pomex that I will prescribe, and my assistant will guide you on how to buy this and how to take this. But before we finish, I'm just a bit alarmed with the thing of memory loss. So allow me to ask you some short questions.
Doctor,
Alexander, can you give me
question. Doctor, just give me drugs so I go.
something? It will be amusing, I believe. Alexander, can you give me the right to share screen?
Can you say again, please? The right for
Share screen. I want to show a test.
just a second. Just a second.
Mr. Victory, this is a test with some questions. You are free to. They are very easy, and you are free to.
You can turn. I'm sorry, Christos. You can turn on your screen now, if you try to.
Yes. Okay. So I believe you can see my screen.
Yes, we can see your screen perfectly well.
No, I can see animals. I'm not an animal.
No. I just want you to identify them, but let's go step by step. At the first the first part of this examination, this is called MOCA. It's the Montreal Cognitive Assessment. This is a test to assess the memory loss, if you have any, and find some ways to make this better. So tell me, you are seeing some letters like A, B, C, and some numbers, and I want you to place them in alphabetical order and in numerical order. Like what is the alphabetical order of the letters you are seeing there, and what is the numerical order of the numbers you are seeing there? Can you tell me? Letters first.
Okay. So I see A.
Yes. Next one.
D.
Are you sure?
D, I'm sure.
Yeah. Next one.
C.
Yes.
And then E. I
E.
think I'm correct. Yeah.
All right. Can you do the same for the numbers you have seen here, like in the proper order from the minor to the major?
Okay. 4, 2,
Yes.
3, 5. Yeah.
Okay.
Correct.
I believe that you can draw this, and you can also draw a clock showing that it is 10 past 11, right?
Showing it is what?
You
10
need to
past
copy
11.
the cube, and yes, and you need to draw a clock which is 10 past 11. Please show this to me. Something very simple, just I want to see a circle and 2 lines and some numbers, nothing else.
Can you see this, doctor?
All right, yes, that's good. And now just tell me the names of these animals. What is this animal?
That is a cheetah.
Yes, like, are you sure?
Cheetah, tiger, all
right,
wild
right.
cats.
This one?
That's a rhino.
Okay, and this one?
That's a dog.
A dog. Like, do you have? Okay, sure. All right. Well, Mr. Victory, we can conclude this test for now. I understand that you are a bit confused, so I would like you to overcome the back pain and the dizziness first. I will give guidance to my assistant, and he will be prescribing an anti-inflammatory and painkiller for the back and a drug for dizziness as well. And I would like to see you back in 1 week to repeat this test with memory, and then we can see how we can work with this. Okay?
Okay, but doctor, what do I have now? Like, what is my diagnosis?
It may be some mild cognitive impairment. It means that either for a short time or for a longer time, you have some problems with memory. This is something that we can address. There are many ways. And currently you have some back pain and dizziness, which are the more acute, and I believe that they can lead you to a panic crisis, and then you think that you also lose your memory. So just make sure that you take the medication to fix this first. Take some rest. My assistant will also prescribe you 1 week leave from your work. So take some rest, eat well, take the medication, and we'll talk again in 1 week. Okay?
Okay, doctor.
Okay, have a nice day. And if you face any exacerbation, please call us again.
All right. Thank you.
Wonderful. Excellent. So now take the opinion of the feedback from the judge. Yana, what is her opinion?
Yana, just now, we will ask her opinion. Just a second. We need to go on YouTube because she's watching it on YouTube and she'll write it there.
Okay.
So does she need to make her opinion like in, for example, the acting of the doctor was 5 out of 5. The acting of the patient was 5 out of 5. How does she have to rate it?
Yeah, 5 or 10, 10 out of 10 for each role.
Okay. So she says that it was wonderful, and now she's going to rate it.
Let's see.
Okay, so the doctor is 5 and the patient is 5, but I think that she used the 5. She meant
5 out of 5.
Yeah, 5
Okay.
out of 5.
And what about the receptionist?
I think that it was about 3.
No, what is her opinion?
I
4.
think I,
4. Your opinion is 4.
Okay. So now each one from you, maybe start from doctor on self-assessment plus on other 2 person. So who is writing 1 out of 5 and sorry, not 1? Oh, that's okay. Okay, so now Christos, what is your feedback on yourself, like a self-assessment?
Well, my feedback on myself would be that it would be way better if we had the patient live, because this needs some specific manual examinations in order to have the cause defined. Also, I should have also asked for smoking history and allergy history before prescribing medication, which I didn't in the course of the discussion. When it comes to the patient, I can't judge because the patient has the right, first of all, the patient faces a health problem and we can't judge their behavior. We only want them to be collaborative. This patient wanted just to take medications and go home. He wouldn't be collaborative in case he would need to stay in the hospital, but yet we can justify this due to his disturbed health condition, and we have to find a way to collaborate with him anyway. And regarding my assistant, he's very helpful. He knows my timetable. He arranged this interview with the patient, and he will also prescribe the medications as I suggested on my behalf. So I'm very pleased with him.
Wonderful. Okay, now Victory, your feedback on yourself and others too.
okay, so like what I played, like the role I played was like, you know, patients do not have time to listen to doctors because they feel doctors like giving a lot of big diagnoses that they don't they don't actually understand it. Our patients do not understand the kind of diagnosis we give. What they just want is they want the pain to go away. They want to just be okay again and they just want to go home. And since it's a telehealth, like what we played was telehealth, most patients are like, oh, just prescribe me something. Let me just go. Give me the pain med so I go. But from what I can say from the doctor, he's actually someone that is down to earth and he actually wants to understand the full picture before giving the drugs to the patient. So that was good. Then for the receptionist, like he was actually jovial on the phone when I called, and then the way he treated the patient was actually okay, because some patients would have ended the call if the receptionist was rude, they will not speak to the doctor. So that's my own take in it.
Excellent. Now, last but not least, Alexander.
So my take, I think that the doctor, he did a great job. He did not say at once his full diagnosis. He made a lot of tests. He even made the patient write down the time, identify the animals, the letters, and I think that that was great, and it was the correct way of doing it. As for the patient, he was all right as well by playing his role, trying to get the pills and the stuff. And my role was kind of small, but I also did my best. So.
Wonderful. So just now, my little bit comments on each one. Overall, yes, 5 out of 5 or 10 out of 10, 100%, very good. A little bit you can improve, start from, okay, maybe receptionist. So when you was asking information, You maybe missed few points. Maybe I miss, maybe you also miss, like a date of birth. For the patient, it is very important to record from the beginning date of birth. And also, like here in our Australia, we have Medicare system. So Medicare card number. Then some people, they have special healthcare card for like a discount or exemption of the charges or that one. Pensioner or any other discount type of cards government is giving. So asking those informations from the patient from the beginning. And also like nowadays COVID time, it need to ask that either you have COVID, those symptoms which need first COVID screening testing rather than directly contacting to the doctor. So because if there is a COVID symptoms, then directly refer to that clinic where COVID test is going on rather than refer to the GP or other doctor. So those type of questions you missed to find out from the patient. And then contact details, maybe you asked, did you ask your contact number or contact details?
Actually, no, I didn't.
Yeah, maybe. Okay, that is also very important because if due to any reason phone disconnect or call disconnect, so how we can contact back to the patient if we don't have the contact number. So contact number is also very important to follow up and enter into the system. Then about doctor, yeah, it was very great. Went into like a real practicing doctor and assessments and went into very detail, but little bit, yeah, as a feedback that like Victory also mentioned, jump very quickly on the conclusion. And the conclusion is not supposed to sometime like or you have maybe loss of memory or Alzheimer's or this and that. So sometimes there are the shocking diagnosis, which is we should also have the empathy with the patient as well. So sometimes that the diagnosis news also become trigger some other post shock of previous some depression or anxiety or some trauma. So therefore it need to figure out that what I can mention to the patient at this moment and what I will mention to the next of kin. So need to ask that who is your next of kin, or maybe the secretary or receptionist can ask from the beginning when they fill up the form, in the form, admission form, when to open the file. It also need to ask from the patient from the beginning that either you have file here in our clinic or not. If have, then first to check the file. And if some information is missing in the file, need to update in the file, like next of kin. And then it need to ask that either same address, same contact number of next of kin, or it has been changed. So if changed, then it need to update in the file. So then some information it need to inform not directly to the patient. In some cases, according to the policy and rules regulation of that country and your clinic and that system. So it need to go through some proper procedure and steps. And also the diagnosis and confirmation that 100% when confirmed that, oh, okay, this problem. So then the serious problem can mention at that time by step, not on the first go. So this was for the Dr. Christos. And for the Victory as a patient, he rolled very well as being a patient that pretend like, oh, I have this one and that one and crazy like a God forbidden real patient. And also gave a little bit tough time to the doctor. Okay, that's fine. One thing, maybe he was supposed to ask from the beginning or at the end, either from the receptionist or from the doctor, better from the receptionist, that what are the charges of your doctor or your clinic? So either I can afford or I can go and look some other doctor and I can't afford. So that will be not so much bill will come after the visit to the doctor. So he either maybe Victor is either very rich, so he did not care about the payment. Maybe he forget this one, or maybe he think that doctor is very generous, Christos, so he is examining the patients free of charge for the sake of the creator. So maybe he assumed that one, which is also fine. Okay, wonderful. Thank you very much for this activity.
Professor
Thank
Kamal,
you.
unfortunately, our time is running up and we are finishing already.
Oh, okay, no problem. Then maybe we can just a few seconds or maximum 1 minute. Any volunteer who can make some creative poetry or poem or songs on anything related to medical medicine or today our this presentation conclusion or summary of all this one. Anyone can from the audience, anyone can say a few words, a few sentences, a few things to summarize what they gain, what they not, what they understand, either it was beneficial or not in a real world, in a real, not like only a fake way.
I think that, yes, there is somebody. Just a second. Let me look if I can tell something and say a few words.
If nobody there, I can recite something of Hippocrates as some kind of poetry, literary mention.
Who can say?
I can.
Oh, okay, wonderful.
Okay.
Go ahead.
Okay, well, I will. This is, you all know Hippocrates was the founder of scientific medicine. He was a Greek doctor back in 500 BC, and he's famous for the Hippocratic Oath that we take at the end of med school. And he's also famous for what is called quotes or aphorismi in Greek. These are very short opinions about the art of medicine, which are the hardships, which are the challenges, and which are the key points to practice. I will read I can't read the prototype in Greek because you won't understand. There is the same prototype in Latin. Probably this is more easy for you to understand, and then I will translate. So in Latin it is ars longa, vita brevis, occasio praeceps, experimentum perillosum, judicium difficile. This means in English that life is short, the art of medicine is long, opportunity is fleeting, experiment is dangerous, and judgment is difficult. And I think that this is something that applies to both research and clinical practice, because compared to the length of work and to the volume of things to be discovered, of diseases to be treated, our life and our time is very short. The opportunity is also fleeting. If, for example, we haven't dealt well with this patient, we would have lost him and his health would have deteriorated. Experimentation is dangerous, and this is the reason we need to validate all research experiments with animal studies and then clinical trials before we can transfer them to humans. And this applies to all the enzymes and drug discovery efforts that were mentioned before. And in all terms, judgment is difficult. Because it is difficult to decide which path to follow. It is difficult to decide which research to prioritize. It is difficult to make career choices, and it is even difficult to announce a diagnosis or set a treatment or monitoring plan. So this is some of medical humanities that we may keep as a take-home message if you want.
Great, thank you. God may give you a happy life here and hereafter as well, and you are the stars of science and the whole global world. And like I mentioned in the beginning, so one day you will do all these scientific more activities and care and this medicine, and to become a doctor, to look after the people and to help them, and try to treat their different health issues. So this is a big job like GCSE was also doing and so many others. So you are doing the noble profession. So have this intention how to like our one is our physiological body and one is inside our souls, our spiritual body. So try to solve the problem of physical body as well as our soul as well, because if soul departure from the body has no value. So similarly, there are so many doctors nowadays who are looking after physical body, but there are very few who are looking the spiritual health as well. So that is also very important to keep balance in the spiritual health and physical health and look after both physical and spiritual health. So if anybody is interested to listen more on this spiritual health and spiritual chemistry, so let me know. It is my pleasure to share more with the spiritual health as well, because now time is finished. So therefore, I don't want to take your more precious time. Thank you very much, and God bless you with more happiness, peace of mind, tranquility, real success here and hereafter as well. Bye.
Thank you, Professor Kamal, for joining us.
Thank you, Professor, from the entire organization committees of this conference that you joined us today for your exciting report and useful information. We wish you a further professional growth and success in science. We hope you enjoyed this webinar just as much as we all.
Yes,
and thank you, Christos. You are brilliant like always. And see you again today at 3 p.m.
Indeed.
Yeah. Christos, due to his motivation and again messages on Messenger, I accepted this one, although I received maybe hundreds of invitations in the last several years, but I am so busy in other things that I did not accept anyone. But due to Christos and his motivation and his again and again messages, I got this opportunity to have interaction with you. So good.
Thank you very much. I really appreciate this, because you normally have invitations from senior researchers and people like them, and you accepted the invitation of students.
Yeah, it is my pleasure to work with all of you. Thank you very much. Thank you. God bless you.
Thank you.
Bye.
All right. Goodbye, everyone.
Bye.
Дякую, що приєдналися до нашого вебінару, друзі. Сьогодні для вас доповідав професор Мухаммад Амжад Камал. Дякую за активну участь та за запитання. Сподіваємось, ця лекція надихнула вас та допоможе вашому професійному зростанню. Нагадаю, що на сьогодні це ще не все. Вас очікує ще 4 вебінари, інформацію про які ви зможете знайти у програмці нашої конференції. Чекаємо вас на наступному вебінарі, який відбудеться об 11 годині. Лектором буде професор Кайдашев Ігор Петрович. Тема вебінару COVID-19, імунітет, гемостаз та неспецифічна резистентність організму, яка є досить актуальною, враховуючи поточну ситуацію у світі. Ще раз дякуємо, що долучилися до